Hello, dear students. Today is a lecture about pathophysiology or pathology, and actually doesn't matter. So, from very beginning, I wanna start. I wanna say to you that it doesn't matter what is it, pathophysiology or pathological anatomy. So, because I'll teach you both, and pathological anatomy and pathological physiology. Let me introduce myself. My name is Knyazhev Igor. I was a teacher of histology, and but also I'll teach you pathology. Treat it like one common subject, pathology. Books to recommend you are Harsh Mohan book of pathology, and of course Robbins book. Sorry. Okay. First of all, I want to share with you what is pathology. Yeah. So what's the purpose of pathology? First of all, try to understand that pathology is a subject which explains. From Greek word pathos means suffering, and actually now pathology is a science of diseases, disorders, and how they are evolved, how they affect our body. And you have to answer three questions during studying of pathology. It's why, how, and what. So how? First of all, why? Yeah. So you need to say so. How it is started. So why it started? Yeah. So every disease possesses its own etiology. Etiology, it's so please Google it if you don't know what is it. Etiology is like the cause of disease, and approximately we divide this into two types of causes. First of all is genetic causes, secondary is acquired causes. These two causes of disorders. Then next question is how. Yeah, how diseases are evolved. That's very tough question. So how disease evolved, and we can learn it on a basic level. We can learn it on deeper level. And my task for in this semester to teach you understand how in deepest way. Yeah, I do understand that it's almost impossible to explain the all disorders so in very details. No one knows it. Even I don't know it. Even Professor Najib or any other professor, no one knows pathology of all of them. But it's well explained in books, at least on middle level. But my task from you to get understanding of at least on basic level of the disorders. When you became a practical doctor, yeah, practical physician, you will learn it. Anyway, you will learn it. But my task is to ease your further learning of subjects. Yeah, because now it's established the first. So pathology is the first subject which encounters you with something with real diseases, yeah, with real pathology, but with real patients, potential patients, and I always said it to students of first year. I always said, yeah, that to understand pathology, you have to learn these three subjects: anatomy, physiology, and histology, and of course biochemistry too. Biochemistry too, it's like additional to understand pathophysiology, pathology is also very important. And But we understand that no one learned it properly. No one. And it's almost impossible for you, especially you had a previous semester in online, and so we always give you discount for this. Yeah. Our first topic is the cell as a unit of health and disease. And here I'll remind you a lot from the histology, especially from cytology part, and something new for you. Okay, so once again, pathology is Greek pathos, suffering, logos study, is applied to modern medicine, the study of disease. So Virchow It's a first scientist, so it's written like Virchow, but it's pronounced like Virchow because it's a German scientist, and ch here pronounced like h. Virchow was certainly correct in asserting that disease originates at the cellular level. But now we know that actually cellular level is not the border. Yes, so actually we have deepest level. It's molecular level of disorders. And that's goal of today's lecture to explain the cellular level of disorders with few correlations with disorders. So that's why if in histology you had a discussion about only normal positions of cells and molecules and activity of some organelles, so here you'll impact with the molecular level of this. Okay, but as written here, so the goal here is to survey basic principles and highlight recent advances that are relevant to the mechanisms of disease that are emphasized throughout the rest of the book lecture. Okay, little fuck up. Okay, genome. The sequencing of the human genome at the beginning of the 21st century represents a landmark achievement of biomedical science. Okay, I hope most of you know what is genome. Yeah, so genome, so if you don't know it, read it here, and here we'll continue. So, in basic words, here, genome, it's a group of DNA, so it's our DNA. So DNA with information in it. But what's the point? Actually, we have approximately 20,000 protein encoding genes. So, and some words they said there are only 20,000 genes in our body, but actually these 20,000 genes are and 20,000 genes are it's just 5% of all DNA. So 1 and 5%. Sorry, 1 and 5% also. So only this information is typical for classical genetics, which you learned in school in biology. Yeah? You learned that one gene encodes one protein. Yeah, and all DNA is to encode these genes, is to encode these proteins, right? But point is that 1.5% is just encoding the protein. But the rest of this 98.5% encoding something else. And it was very, very great, mysterious for us. So when we figured it out, when by methodic known as PCR, polymerase chain reaction, we figured out that we have a lot of genes. We have no idea what the function of it. Till now we don't fully understand all of the genes, but in easy words, we can say that 85% of the human genome is ultimately transcribed, with almost 80% being devoted to the regulation of gene expression. So 80% of genes, 80% of DNA is not to produce something, but to regulate the production, regulate the transcription process, somehow regulated. So let's discuss it a little later. So here is the structure. So might be someone forgot about this. Yeah. So this is nucleus, and inside the nucleus we have chromatin. Chromatin, which is divided into two types: heterochromatin and euchromatin. So heterochromatin is this condensed part of DNA, which usually appeared in peripheral part of the nucleus, and euchromatin, which is present in the middle, usually. Yeah, we have a lot of exceptions, but usually it happens this way. And heterochromatin is an inactive part of DNA. Euchromatin is active part, so which encodes and actively produce mRNA or RNA or tRNA, doesn't matter, but some kind of transcription happens in euchromatin, not in heterochromatin. Okay, but we used to think about genes like chromosomes. So if you learned histology well, so you understand that a chromosome is composed of P arm and Q arm, and it's fused in a centromere, and composed of two sister chromatids. Yeah, so it's one chromosome, and chromosome is condensed only in one process, in mitosis, right? And we can observe the chromosome only in mitosis, in prophase of mitosis, or in meiosis if talking about the germ cells. Okay. Anyway, so if we so the most condensed type of DNA is a chromosome. And if we untie this chromosome, we can see these structures, yeah, with this coiled chromatin. First of all, chromatin, then chromatin is condensed coils, so condensed coils of nucleosomes. And if nucleosomes are condensed very tightly to each other, it's known as heterochromatin. But if it's decondensed, it's known as euchromatin. And it's necessary to what? It's necessary to relieve this part, transcription part of DNA. And this transcription part of DNA is not so simple. We used to think, yeah, that transcription part of DNA is just part of DNA which encodes mRNA, but it's not so easy. So so you see this transcription is shown here. Transcripted part of DNA is composed of promoter, exon, intron, enhancer. Yeah, and why we need it? Promoter is a site of activization. Yeah, that signal to our genes to be transcripted. And then after the promoter, mRNA is produced. So like a reflection of this DNA, and all of this is produced. But then after the transcription, only it's formed only pre-mRNA. After the transcription, formed pre-mRNA. So as it's shown here, so it just directly reflection of DNA. Yeah, you remember this reflection? Yeah, so that adenine reflected into thymine. So in case of mRNA, it's uracil, and cytosine into guanine. Yeah, and guanine into cytosine, thymine into the adenine. So anyway, so it's a reflection. So I hope you understand what reflection. And then this pre-mRNA composed of promoter, exon, enhancer, and exon. So what happens later? Actually, it's pre-mRNA, but actual mRNA is happened after the splicing. It's a splicing, and we learn only exons, and we relieve only exons, and only these exons are responsible for translation protein synthesis. That's my main point. So it's not so easy to say that we have DNA, it's reflected in mRNA, and then protein. No, it possesses this process when some parts of introns, enhancers, promoters are erased from the mRNA to produce proper protein. And actually, genetic disorders, which you learned before, yeah. So we know that disorders like sickle cell anemia, yeah, or phenylketonuria and other diseases from your biological problems, yeah, they are usually it is some eruption inside exon part of DNA. But what if we have some eruption, some lesion in intron part? It leads to nothing. Actually, intron lesions so leads to no pathology after all, and because after splicing, these parts are cut out, and it doesn't affect the protein. Okay, what if happens in promoters? Yeah, it became very dangerous because promoters will activize it. This gene activizes pre-mRNA splicing also, and that's why damaging promoter part. So we'll cut this gene out from our genome, and it makes it invisible. And same to enhancer. Okay, um, so let's here. Oh, sorry. So here it's explained. Okay, we'll explain. The major classes of functional non-protein coding DNA sequences found in the human genome includes promoter and enhancer regions. So promoter, it's an enhancer region that bind protein transcription factors. Transcription factors are usually they are activized by might be innate features, I don't know, might be some biochemical features inside the cell, or might be usually classical example. It's activized by our hormones. So promoters and enhancers are activized by our hormones. For example, glucocorticoids. Yeah, they activize some specific genes, not all genes, but some specific genes. And binding sites for proteins that organize and maintain higher order chromatin structures. Binding sites, it's that I shown you here. Yeah, this part. It's nucleosomes which bind together to form heterochromatin or euchromatin. So because they can't encode some information, yeah, because they are too little. Okay, non-coding regulatory RNAs. So a lot of out of 80% of the genome, they encode the RNA, to control it. So it's not right thing to think that RNA is just a mediator between DNA and protein. Yeah, in most situations it is, but RNA is also active molecule. It's same as active as protein RNA, and this RNAs they can control our gene expression. That's they can regulate the gene expression. Might be activized, might be enhanced. Yes. Anyway, and so RNA. So it's a very deep field which we don't know each of these processes, but you have to know about it. That RNA is not just a mediator between intermediate component between gene to protein. Okay. Mobile genetic elements, also known as transposons. It's very interesting. We'll discuss these mobile genetic elements in immunity system. So this, due to these mobile genetic elements, we can change our genome during our life. So it means that during our changes in our body, for example, coronavirus. Yeah, if you have any new disorder in your body, if you have any new virus or bacteria and whatever, so we have to find the proper protein. Yeah. And as I said before, we have 20,000 genes, protein encoded genes, but mobile genetic elements, they can change it. Some parts of genes, they just jumped from one side to another, and they change the chromatin organization, and they change the mRNA which is produced, and helpfully these mobile genetic elements help to form some variants of proteins, and these proteins are immunoglobulins. Now you know, yeah, we can check our blood for COVID-19 test. Yeah, sorry. COVID-19 test now based on antibodies. It's we check our blood for proteins which force against this virus. Yeah, and how is it possible that different people all over the world they start to produce almost similar proteins against this coronavirus? It is because of these mobile genetic elements which produce specific DNA, produce a specific composition of DNA to help or to form, to encode these anti-coronavirus antibodies. Okay, and then special structural regions of DNA, and they are including telomeres and centromeres, and we'll discuss with them later. So just to show it here, yeah, centromere is the central part of chromosome. It is a non-coding region. It doesn't code any kind of information here. They are necessary only to bind to sister chromatids together. This is centromere, and telomeres are extreme parts of chromosome. So here and here, both of them are telomeres, and the function of telomeres is going to be discussed below. Okay, many genetic variations, polymorphisms associated with diseases are located in non-protein coding regions of the genome, as I said before. So actually our body, so one thing, yeah, here interesting fact that 99.5% DNA is identical in all humans. 99.5, and you see all the variations between you, yeah, so you see different noses, different face, so I mean skin color, different face expression, different so I don't know, noses shapes, yeah, so they are it includes only this 0.5% of DNA. Now, if you're living in one country, it's even this is less. So it's 99.5. It's difference between I don't know, some bushman from southern Africa and I don't know, maybe Indian from southern America, for example. So they're very far from each other, yeah. So and genetically far from each other, and all the changes between them is 0.5% only. And one more thing, there are 99% sequence identical with chimpanzees. You remember this apes? Yeah, these chimpanzees are they are human-like apes, so very similar to us, but still there are a lot of believers that we are not product of evolution. But here it is. So we are different from each other in 0.5%, but each of us is identical to chimpanzee in 99%. So it shows us. I hope no one remains here who disagree with the idea of evolution. I hope. If you think that evolution doesn't exist, so please leave our institute, leave our university, leave medicine, please, because a lot of main principles of medicine are working on principle of evolution. If you don't believe it, so you are not going to be a good doctor, because you're denying the main basis of medicine. Okay, the two most common forms of DNA variation in the human genome are single nucleotide polymorphism and copy number variations. So single nucleotide polymorphism, so from the name you can suggest it's when one nucleotide exchanged to other nucleotide. For example, adenine changed to thymine, or uracil, or cytosine, doesn't matter. So, but one nucleotide changed to another nucleotide. So it's a pointed type of mutation. So its mutation appears not only in germ cells, but also happens in our cells right now. Yeah, in your cell of your skin, in the cell of your liver, in the cell of your bone. Doesn't matter. So anyway, so somewhere in your body happens single nucleotide polymorphism. So when changes happen in one nucleotide. Okay. Let's see it here. Okay, here is a few facts about it. So please read it. So they are still present. Things which are present in our test. We have neutral SNPs. Neutral SNPs means that when these changes happen, but nothing changed. Really, that so because of what? Because one you remember triplet theory, yeah, of gene encoding. So when one of not every mutation, not every single nucleotide polymorphism mutation leads to changes of amino acids. And if it doesn't lead to changes, so it never comes to mutation. It never comes to changes in protein synthesis. So protein will be absolutely similar or with this mutation or without this mutation. And it saves us. It saves us a lot of times and saves our cells a lot of times when this need for mutations happened, and that's it. And we are still alive. And here also, roughly 1% of SNPs occur in coding regions. So that's a little very less, yeah. So when 99% of our DNA is non-coding regions, and usually in these non-coding regions, they are predominantly composed of introns, which will cut out. Yeah, okay, I'll show it here once again. So if mutation happens here, once again, I said it before, but I repeat it. If mutation happens here, so it leads to no mutation, no changes, because this is gonna be cut out by splicing. But if changes happen here in exons, it is only 1% case in exons of protein. So then here mRNA will be different and protein will be different, little different, but it might change the functioning of this cell. Okay, this is SNP. Then TNV, copy number variation. It's a form of genetic variation when some part of genes are mutated. So when we have a lot of genes of similar gene, so one gene is mutated a lot of times. So for example, the first example I remember, so it's happened in polymorphism. It's polyploidia along the okay. Let's do it example. Might be if you were fluent in biology, you had a task, yeah, that this typical Mendel rules, right? Mendel rule. So you have a green pea with a smooth surface, yeah, and you have yellow pea with a shrinked surface, yeah. And if you intermix them, so it's Mendelian rule, so it's spreading of the by Mendelian rule. I hope you remember what I'm talking about. But okay, but what if we intermix? So I mean, you understand, yeah. So intermix two persons with different skin color, yeah. So you know that if parents, so one parent is black, another parent is white, we don't have or white or black offspring, right? They are always brownish. They have different variety of this brown color, but it's never very white. It's never very brown. Yeah. So if we're talking about so parents, interracial parents. So why it happens? Because there are a lot of genes which control the melanin production. Melanin production is localized in a lot of genes. So almost 20 or something. I don't stand on it, but a lot of this is polyploid gene, and chances that half of these genes will be black, half of this became white, yeah, so approximately. And due to this, offspring of these parents will be in shadows, so will be shadows of brown to white, yeah, but it's somewhere in the middle usually, because just by chances theory, so it's will be somewhere in the middle. Okay. If you're not sleeping now, so let's continue. It is important to note that alterations in DNA sequence cannot be themselves explain the diversity of phenotypes in human populations. Moreover, classical genetic inheritance cannot explain different phenotypes and monozygotic twins. The answers of these conundrums probably lie in epigenetics. So, epigenetics is a extremely interesting subject. Epigenetics. So, in the previous semester, I made a conference and asked someone to prepare a topic about epigenetics, and they couldn't do this because epigenetics is very deep and wide subject. Okay, but it's heritable changes. If you remember biology, before the Darwin, we had Lamarck theory, yeah, that during the life genes changed. At that time, Lamarck was being loved. Everyone laughed at him. That say, "Oh no, it's changes during the life. Ha ha ha! You're wrong." Yeah, but now we figure out that actually some genes not changed. Yeah, of course they are not changed. The DNA is stabilized, but sometimes we can control the genes. We can switch on or switch off some different genes. And even to if you have some relatives or someone who you know is monozygotic twins, you can figure out that they are different, right? They are absolutely different from each other. Uh, so sorry, I was interrupted. So we talked about epigenetics. Yeah, and so okay, let's continue. Histone organization. So even though virtually all cells in the body have the same genetic composition, differentiated cells have distinct structures and functions arising through the unique specific programs of gene expression. So in easy words, so you know that in our body we have a lot of different cells and they are very specific. Yeah, for example, cells of our eye is different from cells of our muscle or of our I don't know bone or of epithelial cells. You see, they're absolutely different. And I hope you have been ever interested. So why are they different? Yeah. So why one cell is different from another? So why is their charge to be different? And the answer is epigenetics. So, in other words, so I already said that we have heterochromatin and euchromatin. Okay, I want to return back to this diagram. You see, here we have heterochromatin, here euchromatin. But in every cell, so part of DNA which is in euchromatin position and part of DNA which is in heterochromatin position is different. So euchromatin are the genes which are expressed directly in this cell. If this epithelial cell, it will be this part of euchromatin will be activized. This part of DNA will be euchromatic. But if it's useful DNA for this cell, it's useful for it's useless DNA if this cell is located in heterochromatin. But same useless chromatin which is located in one cell will be useful in another cell. So that's the main point I want to deliver to you in this lecture, that every cell possesses different genes for expression. Some genes are expressed in one cell, some genes are expressed in another cell. And it's tremendously important thing to understand, because I remember my studying time, and when no one explained me this that well, I hope you will understand it right. Okay, so some part of euchromatin. Oh, so, okay, here. Um, okay, here, please read these points once again, but main idea I already said. We have another tool to point the genes for expression and point the genes for to enhance this gene. For example, DNA methylation. DNA methylation, it's a point that we just mark the DNA with methylation. So okay, I will show it to you later. And histone modifying factors. Histone modifying factors. So histone is also a point on which we can influence on. Histone, once again, remind you this from this diagram. This is histone protein. So which binds the DNA. So which forms the factors, and it's understandable that it works not only for binding genes together, binding DNA together. It's also very important to regulate the gene expression. And here it's explained. Chromatin remodeling complexes can reposition nucleosomes on DNA, exposing or obscuring gene regulatory elements such as promoters. So once that's what I'm talking about. Euchromatin expose the gene. Euchromatin. So when histones are getting far from each other, it's exposing of genes. But if we condense the DNA, condense the chromatin, producing the heterochromatin, we obscuring this gene. Yeah, and we obscure the promoters. We close these genes from activation. Okay, then we have chromatin writer complexes carry out more than 70 different covalent histone modifications generically denoted as marks. This include methylation, acetylation, and phosphorylation. And I'll start with methylation process, but we have also acetylation and phosphorylation. So actually, histone residue is marked. So it might be marked. Not only histone, might be some gene itself, some DNA itself might be marked by methylation or acetylation. Okay, let me show it here. So this is histone organization. It is composed of H1, H2A, H2B, H3, and H4. So this is different proteins, but they might be methylated, like this red ring shown here, methylated. So it means what? So if they are more methylated, we have less chance to show this gene out. Yeah, but if it's more acetylized, so you see it forms a euchromatin. We have more acetylated histones, and then genes are more expressed. And yeah, I want to mention this organization. Yeah, H1 is located in parts of DNA which are responsible for expression. Linker histone. It's linker histone which link these histones together. Link this link these nucleosomes together. This is H1. Others are histones itself, which we used to name as histones. Okay. But mechanisms involved in cell-specific epigenetic regulation of genome optimization and gene expression are undeniably complex, despite the intricacies, blah blah blah. Okay, you understand. So, central role in genesis of benign malignant neoplasms. So, unlike genetic changes, epigenetic alterations are readily reversible and are therefore amenable to intervention. So, for example, HDAC inhibitors and DNA methylation inhibitors are already being used in treatment of various forms of cancer. That's the thing I want to say. Why, and that's why we need to answer to the question how, yeah, in pathology. So when we understand how this happens, so why we learn it so deeply, because we can understand the mechanism of it, yeah, mechanism of benign malignant neoplasm formation. And in some types of benign or malignant neoplasms, we can just use inhibitors of these enzymes to provoke these genes, you see, and we can treat some types of cancers. And this microRNA and long non-coding RNA. So let me give it to you in a self-independent work. It will be your self-independent work. So how are they working? That's very interesting. So here we see the diagram which help us to understand the activity of this. So we have RISC complex, but okay, it's already 40 minutes past, and I want to move forward. So this also long non-coding RNA is also in control. Yeah, so I started with it that RNAs which are produced not they are not only to yeah, here's my face. Sorry, non-coding RNA. Non-coding RNA, they also control the expression, gene expression. Not only mRNA is necessary for us. Sorry, some issue with the camera. Sorry. Yeah. Now I wipe my camera. Web camera. That's okay. Long non-coding RNAs is a point of studying. So now we are just thinking about it. So how we gonna use this information? We don't have treatment, but potentially we will get the treatment with the using of this long non-coding RNA. Okay, but it's theory. Let's talk about gene editing. This part is also very interesting part. It's a gene editing complexes. So it's named as clustered regularly interspaced short palindromic repeats, CRISPR. And Cas. It's a CRISPR-associated genes. So with the help of these genes, with its mechanism, CRISPR-Cas, it's a mechanism by which we can edit gene complexes. Actually, it's appeared in viruses, in bacteria, which which force against the viruses, which defends against the viral DNA, because viral DNA can how to say it right, invent inside the bacteria, and it might kill this bacteria. And that's why they just this repeats, they just like checking the DNA. So DNA is it okay or not? Is it changed or not? And if some part of DNA has been changed by viral DNA, so they just cut this DNA out. So why is it important for us? Actually, we can find some specific gene in our body, in our cells, and cut it out and replace it with another gene, same as bacteria did it. We lost this ability, but here in bacteria it's present, and now a lot of genetic treatment is based on this gene editing formation. Okay, also let's discuss it in practical class. So this these are correct mutations and non-homologous joints. Okay, it's continuation of previous topic. So that's how it happened. Yeah, so some part of DNA, so Cas9 protein, so find out this part of DNA which was wrong, and we just find it and cleave it and replace it with some DNA, with some proper DNA. And now we can cut out DNA and insert DNA and donor DNA. And maybe you heard about this genetically modified organisms, yeah? Genetically modified organism, genetically modified food, yeah? So for example, rice, or might be, I don't know, rye, yes, or any kind of soy, yeah. So most commonly it's soy. Some plants which are changed, which we can modify the gene. We just cut some gene out and insert the proper gene, and potentially it will be very important for the humans. We can treat the genetic disorders. We can just cut some pathologic gene and insert proper gene, and it gives us a lot of hopes in the future. Okay, stellar housekeeping. The viability and normal activity of cells depend on a variety of fundamental housekeeping functions that all different differentiated cells must perform. You know it. Yeah, housekeeping. So, what we need to control in our cells? We need to control our pH. We need to control calcium level. We need to control sodium level, potassium level, and temperature. So, and also we need to control our metabolites. So we need to keep our cell in homeostasis. Homeostasis you learned in physiology, yeah. So and you know what is homeostasis, and cellular housekeeping is about this. This is our cell. Here we see mitochondrion, nucleus, rough endoplasmic reticulum, smooth endoplasmic reticulum. You learned it from histology. I'm not going to explain it, explain every of this. I'm going to move forward. New proteins destined for the plasma membrane or secretion are synthesized in the rough endoplasmic reticulum and physically assembled in the Golgi apparatus. Smooth endoplasmic reticulum may be abundant in certain cell types to produce hormones, lipoproteins, and might be form the might modify hydrophobic compounds to make it hydrophilic compounds and to excrete it with our kidneys. Okay, and cells catabolize a wide variety of molecules that they endocytose, as well as their own repertoire of proteins and organelles. Okay, you know it. Let's move forward. What are proteasomes? So proteasomes are specific organelles, but it's hardly to we can name the organelles. It's just composition of proteins, which are enzymes which kill other proteins, which destroy other proteins. If I said you in the beginning, yeah, that when proteins are produced wrong, for example, due to some genetic changes or something else, or might be due to some physical or chemical damaging of this protein, now we need to degrade this protein to produce a new protein. Yeah, and proteasomes is a function is this. Okay, lysosomes. Lysosomes are intracellular organelles containing enzymes, and they possess acidic milieu inside, and this acidic milieu activizes enzymes and can digest our own cells, cell components, and they can destroy macromolecules like proteins and polysaccharides, lipids, nucleic acids. Doesn't matter. So we can digest everything, almost everything, by lysosomes. So it's like a hard type of defending of our body. Okay, and peroxisomes. Peroxisomes are specialized cell organelles, and they're predominantly present in neutrophils. You remember neutrophils in the blood? Yeah, they're a defending organelle, which actually destroys cell membranes. Because so we'll discuss it right now. It's next. Some a little later we'll discuss it. It forms a radical oxygen species, which helps us to kill bacteria. Okay. Contents and position of cellular organelles also are subject to regulation. Okay, you know it, read it, so it's nothing new for you here. If you learned it properly in biochemistry, you know it. If not, so repeat it here. So growth and maintenance require a constant supply of both energy and the building blocks that are needed for synthesis of macromolecules. So we need energy, yeah? Okay, that's it in this lecture. I will continue this lecture in this week. I guess I write this lecture down right now and send it in a blog. So next time. Okay, thank you. See you next time.